And thank you, Amy. And so I'll just, we have such a lovely turnout, and I think a testament to our speaker and the continuing legacy of our speaker today, to see folks coming to join us today, and old friends. I see Patrick O'Grady's joined us, folks coming back to be part of this. And so I'll introduce myself quickly, since we have a really broad group of folks. I'm Justin Brashares. I'm a professor in environmental science, policy, and management, and I'm the organizer for this semester of the Wildlife and Conservation Seminar Series. And I wanted to quickly introduce a graduate student, Amy Fingerly, who's here with us today. I believe Amy is in the Carlson and Grantham labs, and Amy has, we've tricked Amy into serving as the wildlife program coordinator for this semester. And in that role, Amy is helping us organize the seminar series and really helping us as a community chart our path forward. So I want to thank you, Amy, and introduce Amy. And I'm also going to ask Amy if she wouldn't mind to put her email in the chat for folks who might have questions about the seminar series. We are operating on a virtual basis, so all of our talks this semester are on through Zoom, and we have scheduled speakers every other week. Okay. So with that, I'm going to go ahead and just declare my my tremendous pleasure in introducing a colleague and friend, Professor Vince Resh, today. And I could easily spend all of our time together, the next 50 or so minutes, relating components of this of the illustrious career that Vince has had and continues to have. I'll point out, as he continues to publish actively and continues to reach souls and minds actively. But I'm not going to spend the next 50 minutes talking about the hundreds of peer-reviewed papers in evolutionary ecology, in systematics, in ecology of primarily freshwater insects. I won't talk about all of the work that has gone so far beyond our campus to impact human health, development, poverty, or so many other ways that Vince has connected with the issues of our era and had profound impacts on some of the great challenges we face as a species globally. And hopefully Vince will talk a little bit about those things, but I encourage those who don't know about the legacy, and again, continuing legacy of Vince's work, to spend more time looking into that. I want to talk quickly about some of the things, the other things that have made Vince so special to me and to our community. And those are the ways in which he has touched so many lives through his friendship, his mentoring, and his commitment to our campus and beyond. And I think any of you who have interacted with Vince will know, and some of my favorite interactions with Vince are, whenever I approach Vince, I know that when he asks how I'm doing, it's not going to be a rhetorical question. And in fact, Vince will usually he'll cross his arms and lean against a wall, which is a signal to say, you're not getting out of this easy, buddy. Let's hear it. And I'm so grateful for Vince's sincerity as a mentor and friend, and someone who truly cares about the fate of those around him. I'll also say just a quick anecdote, and Vince will probably get mad at me for outing him on this, but early in my time here, Vince invited me out for lunch, as he has several times, to just provide support and mentorship. And towards the end of our lunch, Vince looked at his watch and said, I gotta go. And I said, oh, okay. And as many of you know, in a community where everyone is, if they don't have really important things to do, they generally pretend they have really important things to do. And in this case, Vince said, yeah, I gotta go. I want to catch the matinee. And I kind of looked at him, and I thought maybe he was joking. He said, oh yeah, every Thursday or whatever day it was, I catch a movie just by myself, at like 1 p.m. in the day. And I just thought that was so fantastic. And I'll be honest, it gave me hope that there is a future in this job and in this life where you can go to a matinee once a week and just say, and I just love that Vince was, for someone who's accomplished as much as he is, was so open about the fact that, like, yeah, I'm going to go sit in a dark movie theater in the middle of a day. And it was just fantastic. So Vince probably didn't even realize you were inspiring with that move, but it was very inspiring. I'll also just end by saying it's very rare that I can say, or anyone can say with honesty, that the talk we're about to hear is being given by the best person on the planet to give that talk. And that is undisputed in this case. Vince joined Berkeley in 1975. And as I said before, has both profoundly influenced and shaped a variety of disciplines, but particularly the work and the legacy and impact of freshwater science on this campus. Really, yeah, it's special to have you here, Vince, and we're so grateful for your being here and sharing this history with us. We all recognize academic settings, and particularly ours, have an amazing ability to ignore history or to move on very quickly. In some cases, to repeat exactly the same science. That's a little bit dark, but that's the reality in some cases. So we love having you here and to remind us of the really important legacy and history of freshwater science on this campus.
Thank you, Justin. Thank you, Amy. In terms of the last story you told, for several years, I taught a course called Science in the Movies. So actually, I was really leaving for a matinee to prepare for class. That was always the justification I used, although the students in my lab never really believed that, as I would sneak out. So what I'm going to do is try to encapsulate some of the real highlights of what I think has happened over the last 150 years in aquatic ecology at Berkeley. And I literally mean 150 years. But to start that 150-year story, we're going to really talk about the first part of the history, and that's about Strawberry Creek itself.
Hey, Vince, I'm sorry to interrupt, but you'll want to restart sharing your screen. I turned it off so we could all see each other, but if you'd like to restart that so we can see your slides.
Here. Okay, is that it?
Perfect, thank you.
Okay, good. So what we're going to do is we're going to really start talking about Strawberry Creek. And then we're going to really move into the personalities that have shaped aquatic ecology at Berkeley. And I have to say, I'm going to stick for the most part with people that have been retired. That's a safer group to use, but I will end talking about some of the current things that are going on. So with that, let me start the slides. And first of all, let's go back and look at the history of Strawberry Creek. This is a really big issue of I think I skipped one right here. Yeah, with all of our, almost every seminar, or even if we go to a play, they open up with talking about this is the land of the Huichin-Ohlone people. And in fact, we have Native American settlements here according to Kent Lightfoot over in Native American Studies, probably for the last 10 to 12,000 years. But the Huichin people were actually clearly on campus. They used the campus seasonally, and they actually had it at the faculty glade. They had a campsite has been found where they would probably were harvesting salmon. But of course, the main group was down at the Shell Mounds, which is in current Emeryville. Well, in the late 1700s, we see the Spanish explorers and pioneers coming in. And again, one of the tragedies, the Huichin people were moved to Mission Dolores. One of the things that's kind of fun to look at on campus, if you walk down towards Center Street, you'll see this little plaque that I have in the lower right-hand corner. This is one of the sites that the Spanish explorers actually used the term the Golden Gate. Remember, the Golden Gate wasn't named after the bridge. The Golden Gate was this beautiful area they saw, especially at Christmas, where the sun would set right in the middle of what's now San Francisco County and Marin County. And this is really the origin of that. And there were a couple of other places that they did this, but this is one of the main ones. You can't actually see the Golden Gate. There's a couple of 10 and 12-story buildings that are blocking it, but that was actually where they started it from. Now, one of the things that's interesting, and these are a couple of sites of what Strawberry Canyon probably looked like, at the early part of the campus or before the campus was settled. But if you go on San Pablo Avenue, you'll see a series of monuments, one of which here says the northern boundary of Rancho San Antonio. One of the things that was interesting about the way they described landmarks, they described boundaries, was they used the creeks because these were the most visible ones, especially in the alluvial plains that we see that come down from the hills. And this was actually, like a boundary between what we call El Cerrito, which used to be called Rust, and Albany. But of course, we have California statehood, we have the gold rush, and a variety of other things that are going on before the campus was actually here. But all of these things were strongly influenced the campus. If we look at the University of California, the origins actually started on land that was purchased by a retired sea captain that lived actually had a small cabin on the campus. But the land that was sold was actually the lower part of the campus. It didn't include the hills. That had to be purchased later on a very, very complicated land exchange, because one of the things they wanted to make sure they had a reliable water supply, and part of that reliable water supply was the springs that came on the campus. And of course, they needed reliable water, they needed potable water. And this quote was actually in 1887, is a very, very known quote of why they chose the campus here. Because originally they thought about going to Napa. That was one of the places among others. And here we see all other striking advantages of the location. Could not make it a place fit to be chosen as a college home without this water. With it is excellence of double value, 1887. The problem is, when you look at what was going on, the problems that we see today of a variable water supply in Strawberry Creek are actually the same as was occurring 150 years ago. And so the title of the 150 years of history on the Berkeley campus of aquatic ecology really goes back to 1873, where the first fish hatchery was located actually on the campus in the North Fork of Strawberry Creek. The problem was there was too variable a water supply. There really wasn't enough, so they moved it to San Leandro. And there they continued it for many, many years. They also did one near City Hall in San Francisco. And if we look at those features, we see that it was, fairly small. They had a series of channels, perhaps 50 channels, and there's no records about how much fish was produced. But what was recorded was the fact that the fish they produced were not the native rainbow trout. Typical of what's happened in terms of introductions, they brought in brown trout and brook trout. And somewhere I actually saw a reference that they may have even brought in corrigonids, whitefish from the Great Lakes region. But of course, all invasive species, all introduced. And eventually, of course, the rainbow trout that went throughout the world was from the McCloud River drainage. These acclimatization societies were very interesting. What they were trying to do was to make the area appear like where people came from, especially from the East Coast. And so that was one of the reasons that they wanted to bring the brown trout and the brook trout in. Now, Strawberry Creek actually features very strongly in art. There was a very well-known California artist whose name was Edwin Deacon. And among his series, there's a huge collection of his at the Crocker Art Museum in Sacramento, but one of his series was of Strawberry Creek. And they all have kind of a dark, mysterious quality of them. Interestingly enough, the paintings are owned by the California Department of Finance. And I've never really been able to figure out or find out how they got them, whether they were for unpaid taxes or whatever. But there's a beautiful collection of his work on display at the Crocker Art Museum, which is just a beautiful train ride away from Berkeley. Now, unfortunately, we get to 1895, and that the campus is of double value because of the water. Look at this. It was a junkyard. Raw sewage was poured into it. And I think this was probably the beginning of when you started having problems with the salmon, especially the steelhead, running up into Strawberry Creek. It was just the pollution that was coming in, and the fact that this was just so cluttered with debris. Now, when you look at the campus, I think all of you know there's probably two forks. There's the North Fork, which runs by as the Chancellor's House and comes off the that Hearst Avenue neighborhood, the North Berkeley neighborhood, and of course the South Fork, which is entirely on the campus. But there was a Middle Fork, and the Middle Fork was filled in actually by mandatory PE classes. For 4 years, the colonel that ran the PE program at Berkeley would have the students, mostly male students at this time, were going out and hauling dirt and to fill in that Middle Fork. And the reason they did that, they wanted to create a track stadium. The colonel was very interested in running as an exercise, and so the track stadium was put in where that Middle Fork is. And the giant eucalyptus grove that was built related to the track stadium as well, because what they did was they wanted to make sure that any records that were established that wind wasn't a factor, so they put that in as a windbreak. When they were building the VLSB annex, the addition with a molecular biologist, they found that there were stairs that were leading actually to old locker rooms that were related to the gym and to the track stadium. Now, where does Strawberry Creek go? Well, the outfall is actually at the Berkeley Marina. The Seabreeze Cafe, which is a little gold mine that sells overpriced sandwiches, is right there, and you can see the outflow. And what's interesting is some of the fish, the hardy ones like sticklebacks that could withstand salinity, actually established a colony which originated from Strawberry Creek that were washed down through the culverts. Now, one of the big things that happened in the Berkeley campus was building Memorial Stadium, the football stadium. And this has had long-term repercussions. So in 1922, they decided to build a stadium at the base of Strawberry Canyon. And there was a lot of opposition to this. Not just the people that live up in the canyon, which sort of object to everything, but really they thought that it should be down below, it should be on the campus itself. And John Galen Howard, who is the great campus architect, he and Berkeley had a parting of their ways over this, because to him, this was one of the most famous bird watching sites in the Bay Area. And especially the way they dug out the stadium. What they did was they used monitors with high pressure water. In other words, they dammed up Strawberry Creek. They used the hydraulic pressure to build that essentially that hole in the side of the hill where the stadium was. And what this did, it actually just created a huge sluice of mud and debris, that if there were any fish that were migrating up Strawberry Canyon, this ended it for sure. And this actually was sort of the beginning of the town and gown problems at Berkeley, because people really in the city were really opposed to the fact that they had dug this out. And as I said, the great architect left over this issue. So if we look at the kind of the issue of water pollution and regulatory agencies, the culverting started in the 1800s, and today Strawberry Creek is culverted, with the exception of Strawberry Creek Park, all the way down to the bay. There was increased erosion. Remember, there was raw sewage going in, there was cattle grazing, and this really led to just the further degradation of the creek. And then in the 30s, there was a big fear of a cholera outbreak. Cholera was breaking out in several parts of California. So the city was very concerned about open creeks, and they really pushed to have the creek actually covered over its entire length. And then this, and the part that I remember very, very well, in the 1980s, the city of Berkeley actually advised against direct contact with the creek. And as you know, if you've ever been there on weekends, kids are playing in the creek all the time. And they actually were a series of signs put around telling people to avoid the creek. This reached a peak when the coliforms, E. coli, which are not generally toxic in themselves, are pathogenic in themselves, but they're an indicator of viruses and other pathogenic. The levels in Strawberry Creek were reaching 500,000 per 100 milliliters, where really you would be expecting in an urban creek like this, at least in the 1980s, from 100 to 1,000. And this was really a huge problem the regulatory agencies were very, very concerned about. The campus was really concerned about potential fines and just, another set of bad publicity about what was going on. Now, I do have this little picture down at the bottom because Strawberry Creek was very famous because about 50% of the lab rats that are used in the world for research are the Long Evans laboratory rat. And in 1915, Long and Evans captured a wild Norway rat in Strawberry Creek, and they used that to start the breeding line, which today is the most widely used. Millions and millions are used every year for laboratory research, and its origin was on the banks of Strawberry Creek. So let's go back to these high coliform counts. 500,000, just unbelievable. I mean, the smell, the water would have a kind of a whitish color from the beginning of sewage treatment. And one of the things that really got the campus, the faculty and the staff, and Joe McBride would remember this very, very well, is the campus said, this is not a problem of ours. There's dogs that are running loose on the campus, and the dogs are pooping all over the campus, and it's getting into the creek, and that this is really what the problem that's producing these coliforms. And their solution was sort of a typical administrative solution. What they did was they passed a leash law. So people had to have their dogs on leashes. And of course, this was absolutely insane. I mean, this made no difference at all. And in fact, what the problem was due to was the fact that the football stadium was built on the Hayward Fault. I mean, and this was another objection that they had, where they said this is not the place to build it. Now, California is unique in really compared to most of the United States, in fact, most of the world, in that we have two systems of sewage. We have a sewer system that basically takes waste, takes toilet discharges, and then we have a stormwater system that takes runoff water. And due to the fact that the Hayward Fault and the causing breaks, both the sewer system line and the stormwater line cracked, and they were right next to each other, and that they were put in the same lines under the stadium. So what happened is the 6 times or 7 times a year when Berkeley was playing football, and people would go to the bathroom at halftime, and they go to the bathroom at the end of the game, this sewage line had such a surge in it that it pushed the sewage out, went into the storm sewer, and that was carried because that all went into Strawberry Creek. So that was the real problem. Now, this actually really was the beginning of the restoration of Strawberry Creek, and it involved faculty, it involved staff, it involved students. And basically, we see a research component right at the beginning when they repaired the sewer lines under the Memorial Stadium. They sealed the sewage lines, but they left the storm sewer alone so they could look at further movement on the Hayward Fault over time. So even with this, we see a very strong movement towards research. But all of a sudden, I mean, all kinds of other problems were clear with Strawberry Creek. You look at the fact that the way the walls of the creek, the banks, were lined. I mean, there was just flashiness, and the flashiness, of course, led to floods. The floods led to problems on the campus. Here you see in the eucalyptus grove this massive amount of water that was coming down during floods. So what ended up happening is that I think we could trace a lot of this to a graduate student, Bob Charbonneau, that was a student in environmental design, that decided for his thesis what he was going to do was look at how we could come up with a restoration plan for Strawberry Creek. And one of the first things, he involved a lot of faculty, involved a lot of students. And, he started putting information together. Like there was a report that steelhead were seen in Strawberry Creek in the 1930s. That was the last time they were found. Well, to me, this is probably an urban myth because, if you think of all of the silt that came in from the construction of football stadium and all the other impacts that it had. It's sort of hard to believe that steelhead were running up there in the 30s. But for sure, when fisheries biologists were actually starting to look at, no fish were found in the 60s to the 80s. Now, this is in Strawberry Creek. If you look at like some of the fountains on campus, there were turtles that were thrown in and goldfish and things like that. But in terms of the creek itself, no fish seemed to be get established. And, if we looked at the aquatic insects, the fish foods that were there, they 15 to 20 families of these very intolerant species, pollution intolerant species were found in the canyon. You looked on the campus, they were 5 family, and they were all very, very pollution tolerant. They were fly larvae and worms. So the idea was, let's start by looking at the soils, the geology, the land use, the storm drainage pattern. For example, water hits that ChemAgro lawn, and 15 minutes later, you end up having a peak in the creek. It's very, very flashy. And then eventually to develop catchment management and the big issue, of course, was the funds. And what was really interesting about this is that this was really a combination of faculty, staff, and students. So Bob Charbonneau I mentioned, but staff, Carl Hans and Sonia Bjorn Hansen. Tim Pine is the kind of the current guardian of what's going on in Strawberry Creek. But faculty such as Joe McBride were very, very involved. And actually, they formed a Strawberry Creek committee that I know Stephanie has been involved with, and Matt Condolf and other people as well, that really oversaw what was going on in the creek. And there were a lot of problems. There was, point source pollution. There was non-point source pollution, broken sewer lines that ended up having raw sewage going in, and water mains that broke. And of course, one of the big problems with water mains is after the 1980s, East Bay Mud started using chloramines instead of chlorine. And the chloramines were toxic to fish, so that if you had tap water going into the creek, you ended up having fish kills. The same was true with the pond, and the Japanese pond in the botanical garden, which was releasing tap water. That's how they filled it. That's how they kept it going. But there were misconnections of sewer lines and storm drains. And, at the swimming pool, when they take the water out, they just dump it right into the creek. And, you look at the concrete walls here. There's no habitat that you're going to have for aquatic life. And there were sort of bizarre things that happened, like at the faculty club, which Keith is now the chair of. When they'd be frying French fries for lunch, they'd have all the grease that was left over. They take it out and they just dump it in the creek. So there was just a lot of crazy things that were done that you wouldn't think in the 80s with the environmental concerns. But all these things were basically had to be dealt with in terms of keeping Strawberry Creek going and eventually making it into something that was going to be a restored site. So, the hydrology was looked at, and, especially structures, the hard structures like bridges. When they collapsed, they were really the solution was these biotechnical approaches. And I'm going to show you one in a minute, but, gullies, repaired so that you wouldn't have siltation going in, rerouting, cross connections. And the big thing, of course, was education and outreach. Going to the faculty club saying, look, you don't want to dump the fat in the creek. And this, of course, is a continuous issue. And Tim Pine with the H&S it really does a marvelous job keeping students involved. And this has been really, really quite good. Here's the crib wall that I'm talking about. And unfortunately, it's been so successful and so overgrown with vegetation. But this bridge was collapsing. And for about half the price, they could put in these cut redwood logs, plant vegetation, and put things like notched weirs in, not metal, not stone or not cement ones, but to build up aquatic habitat that you could end up having a place for insects and fish to use as refuge during floods. And so this was really a great project. And I remember several of us, Joe McBride and I and others that were involved, Matt Condolf, we wanted to come up with a plant list of what to have. And of course, there was no similarity in any of the lists we had. And I remember Joe McBride said, to me one day, plant diversity is not alders, you know, which is what I wanted to put in for nitrogen fixation. So we planted everything in, and of course, you know, the plant ecology worked out, and the probably the most plant diverse spot in a small area in California ended up, you know, some species thrived, and of course others died off. But again, it's a demonstration project. It's still being used. One of the keys about demonstration projects on the Berkeley campus, they always look good. That was sort of the idea that, you know, people from the cities or from the different agencies would come and look at, and they were really nice. The little filtration marsh that drains the Dwinell parking lot. Again, looks nicer than it has to, but again, it makes this idea appealing. And in the case of the redwood crib wall, the way we see it mostly used now, and they're used all over California, is they take giant boulders and eucalyptus logs. So they really aren't pretty at all, but it's the same principle that this demonstration project had with Strawberry Creek. I will conclude with fish introductions. You know, we've tried a lot of different fish in the campus. I remember once the suggestion was we actually even tried to bring some trout in. And I remember the administration response was, you know, somewhat typical. And what do we want? You know, the homeless fishing on campus. And so we didn't do that, but instead we went with fish like the minnow, the California roach, or the three-spined stickleback, which are very, very hardy species. Sacramento sucker were in there. We haven't seen them for a while, and I'm not sure if they are, but you know, we actually documented all of the places where the fish were brought in so that we could look at if genetic studies were done over time. At this time, everybody was doing electrophoresis. It was pre-molecular approaches, but there is documentation. Most of the species were brought in from Redwood Creek in Oakland, and of course we had, you know, herons and egrets, which are fish-eating birds that became established in the creek. A lot of education, you know, the stenciling that was done was very interesting, because originally it was just with a stencil and paint, but you know, it was done in English, and then it was done in Spanish, and then it was done in Mandarin, and it was really you know, kind of got to be a great thing. And then eventually we got to these rubber labels that were done, and producing actually a walking tour, which is still available. Now it's, instead of having it in paper, it's online on the Environmental Health and Safety website. But, you know, sort of the last time we looked, you know, over 300 students have Strawberry Creek as part of their education experience. And these, of course, are the general biology students, where we have a lab that deals with Strawberry Creek. The environmental science students, actually even art students, those that take the water course that's in earth science, you know. And so it's really a great thing. And plus, you know, we've had a lot of monitoring that's been done by students. So a few years ago, when we had a big oil spill, we had a lot of pre-data. We could get post-data. And in fact, that was very instrumental in getting a large fine that against the polluter in terms of going to a natural resource enhancement. But, you know, ongoing problems, siltation from construction. But, you know, the restorations continue. If you look at the eucalyptus grove, it's sort of fenced off right now, but they're doing a crib wall there instead of just replacing the concrete. And I think, you know, this has really been very good. And, you know, Strawberry Creek is a restoration site. It's been written up in the New Yorker and in scientific publications, mainly because of the demonstration projects that have been done there, which are very, 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 very good. All right, now let's switch and let's start going to some of the personalities that have been at Strawberry Creek. Sage Hen Creek Field Station, Rob Brew is the current director. Don Ehrman was the director. Before that, Jim Kirshner. Really a great series of modern directors. Strawberry Creek, it's Sage Hen Creek, it's located up in the Sierras. And it was funded on kind of an interesting arrangement. The legislator was giving money to UCLA to set up a center for marine studies. And Berkeley said, you know, let's have one for freshwater studies. Let's do it in the Sierra. And it got funding to purchase land and or lease land, I guess is the correct term, and start building some buildings. Now, the first director was Paul Needham. Now, any of the students that worked on aquatic insects would know the name Needham because his father, James G. Needham at Cornell, was probably the most, well, it was the most famous aquatic entomologist of his kind, especially famous for his work on dragonflies. And that's him over in the right. Now, his son, Paul, went to Cornell, where James G. was teaching. And then after that, his career was very interesting because he went into government service for a long time. So he was at Stanford in the Bureau of Fisheries Station there. Then he was at Convict Creek with Fish and Wildlife. Then he went to Oregon as the director of fisheries. And really, so this is like 16 years later, starts as a professor at the Berkeley campus. He's a very interesting guy. In that he's, a fisherman, very, very much interested in the outdoors. But, in this era where, students will talk about, issues about graduate student rights and everything, Needham did something that was pretty bizarre, even for that time. None of the students that did dissertations were allowed to publish their research. Needham was intending to have a giant collection of everything in a book that he was going to write about trout in Sage Hen Creek. He died in '64, and various postdocs like Dick Gard really got all of the former students that had worked there to publish their research. Because it really a lot of that never would have gotten out, and again, it's sort of hard to imagine. But I remember when I was here, first came in the early '70s in the old zoology department, the students were not allowed to publish their dissertation research until their dissertation was filed, with the argument that a dissertation is original research. So that, of course, has changed since then. But again, very, very different attitudes about publications and students at that time. So I have to talk about Robert Usinger. Robert Usinger was probably the entomologist of all time. He was an aquatic entomologist, but he also worked on bed bugs. He kept a colony of bed bugs in old mine adits on bats, and when he would bring them back to the lab for his studies, he would put them in a little jar and then put them on, tape them to his side so the bed bugs could feed the whole time that he was driving back. And he wrote this marvelous autobiography, an entomologist, but also wonderful books on rivers and streams in Sierra Nevada natural history. And his specialty with aquatic insects was the true bugs, which of course are related to the bed bugs, such as the bat bed bug and the human bed bug that you see there. And of course, bed bugs, the reason he could do this by putting them on the bed bugs to feed on his side is that they're the only blood-sucking insect that doesn't transmit any disease. It's never been shown to do that. So Usinger was really just a great entomologist, and from everything I've ever heard, a great person. Now, his big contribution was the aquatic insects of California. And, I found, especially when I was working in Africa, every lab that I went into in Africa, this is what they were using for their identifications. I mean, it was the first book to come out really that had usable keys, that was broad at the family level. And his specialty in it was the Hemiptera. But what ended up happening was he got the typical group of academic authors and graduate students to write it, but there were three that really were remarkable to me, and these are the first three. Bill Day was an executive at a big advertising agency in San Francisco, and he came up with the idea of those of you that are wearing Levi's, the red tag on the Levi's. That was the identification, that was the marketing or the brand that he came up. So executive in advertising, no background in ecology or aquatic insects other than he was an avid fly fisherman. He had a heart attack, and his doctor said, you got to do something to get your mind off of work. And so he converted a bathroom in his house to rearing mayflies, described some new species, and actually wrote the mayfly chapter for the Aquatic Insects of California. Don Denning, that wrote the caddisfly chapter, was a salesman for Chemagra, a pesticide company. But to me, the most interesting was Bill Bentenik. Now, he wrote the chapter on black flies. Now, he was a graduate student at the time, but he developed a severe allergy to laboratory alcohol, leaning over, sniffing the fumes from while identifying specimens. He ended up quitting, quitting science. And what he did was he started to use fiberglass in building sailboats, and then went into using fiberglass to build these huge multi-million-dollar yachts, and was just incredibly successful, really very, very wealthy. And I talked to him years ago, and I was talking about the aquatic insects in California, and he said to me, and this is a guy that had such a successful career, he said, the biggest mistake in my life was leaving aquatic insects. And I just found that fascinating, because you look at all he accomplished. Now, many of you that are working with fish know about the aquatic insects in North America, the Merritt and Cummins and Berg, and it's now in its 5th or 6th edition. Originally, they wanted to simply redo the aquatic insects of California, but they couldn't get a lot of the old authors to agree, because they wanted to bring in more modern authors. So they ended up having an independent publication to do that. But again, that's now the widely used site. But you still see the aquatic insects of California for sale, especially like on Amazon as original copies. Needham and Usinger did an incredible study that had long-reaching implications even to today. They did it in 1956, and they published in a journal. It's actually published by the College of Natural Resources, called Hilgardia. And it was called The Variability of Macrofauna of a Single Riffle in Prosser Creek, as indicated by the Serber sample. The Serber is simply a square-foot sample. What they did, they got 4 graduate students, and they went up to Prosser Creek. Prosser Creek, that area they studied, is now under a reservoir. And they took 100 samples in a grid. And they, of course, measured flow direction, depth, things like that. But what they found when they did all the identifications, they brought a statistician from what was then called the Lawrence Radiation Lab, or the RAD Lab. Now it's called the Lawrence Berkeley Lab, who had a great name for a statistician, Evelyn Fix. And what she did was she analyzed the data from these 100 bottom samples, and she came up with figures that were just astronomical. That to get significant figures on wet weights, 194 samples were needed. Just to get numbers, 73 samples were needed. So what this really just, even when these figures were corrected by people that were looking at that, because they published the raw data, it really put biomonitoring and pollution studies back, because even today, when you look at most pollution studies, they're taking 3 to 5 samples because of the idea, or even sometimes 1 at a site, with the idea that the variability is so high that you just can't deal with it at all. And this is unfortunately one of the things that's happened since then. If we look at other aquatic people at Berkeley, John Anderson, who worked on mosquitoes and a lot of parasitic flies on wildlife, is primarily a veterinary entomologist. When he retired, he moved to Bend, Oregon, and wrote this great book called Fly Fishing. And if you can see the subtitle, he looked at insect drift and fish diets. It really is a scientific approach to angling. Hal Daly, that's in the center, was very interested in fly fishing, and he and I actually taught a fly fishing entomology, or angler's entomology, through the Golden Gate Angling Society, and actually raised quite a bit of money towards the restoration of Sagen Creek. Dick Garcia, very, very famous mosquito ecologist, also at Berkeley, worked on aquatic insects, shown in this bottom slide here. No, that picture is not upside down. These are called back swimmers. That's literally how they stay. They use that unique part of the aquatic environment, and they sting very, very. Their sting is very, very painful. So, if you look at since 1975, since when I came, we've had 42 graduate students, and they've all done very well. They've gone into industry, they've started consulting firms, in academia, to government service. And I think if there was one theme that they all had in their dissertations, was this quantitative approach to natural history. In other words, doing very traditional studies but doing them quantitatively, and especially doing basic research and to answer very, very applied questions. So, biomonitoring, mosquito control in wetlands, restoration, my involvement in river blindness for 15 years in Africa, and the Mekong for another 10 years. Really, what we were very, very pleased, we get so much long-term data because we had so many undergraduates and graduate students that in 2015, we published put all of our data on websites. So it's available, and we've seen dozens of papers have used it because it really was the best consistent data set, because I took all the samples and the same graduate student did the identification at one of the sites at one of the UC reserves, the McLaughlin reserves. And now really our main efforts is working with former students, former postdocs on modeling landscape effects on population genetics, which I want to thank Patrick O'Grady for getting me, explaining to me as a novice. So if we look at the graduate students, these are various times they've gone on. Although it's too late for my career, we now have an in at NSF and ecosystems, and that the ecosystems program, which is a huge program in biology, is run by 5 directors, and 2 of the 5 are out of our lab, Justin Lawrence and Gary Lamberti. So like I said, it's too late for me, but it's wonderful to see how successful they are. Other faculty members, Don Ehrman was here, came in the early 1970s, and then he was a very, very good administrator. He worked on trout at Sachem Creek. He was the director after Starker Leopold. And his wife, Nancy, worked on caddisflies up there, especially a very interesting, unusual caddisfly that actually has part of its life cycle as a larva in terrestrially feeding on terrestrial vegetation. And then George Barlow, who was really the great cichlid biologist. And now, of course, we see Chris Martin is doing some of those same things. But George really was a marvelous guy. The picture of the person on the right is Dennis Newbold. Now, he was one of Don Ehrman's graduate students. And he went on to, first of all, to Oak Ridge and then to the Stroud Lab at the Philly Academy of Sciences. But his big contribution, which is very, very important today, was the idea of nutrient spiraling. And he got that from working in the Sierras. There were a bunch of civil engineers that were really very, very strongly motivated to ecology. Pearson and Jerry Thomas, they were essentially wastewater engineers, but really started looking at things like diversity indices and the rivers going into the bay itself. David Jenkins, one of the great water chemists for wastewater. Hugo Fisher, a modeler. Hilgard Sternberg, not an engineer. He was in the geography department, but was the expert on the Amazon. Luna Leopold had been chief hydrologist at USGS, came to the faculty. Jim Kirchner, really an excellent teacher as well as an excellent biogeochemist. He left and went to Switzerland, but stayed as the director of Sachem Creek for 4 years. And of course, Bill Dietrich recently retired. I was lucky in having a project with him for several years on the Klamath, and a very, very wonderful guy to work with. If we look at one of the offshoots of the engineers at Berkeley, and it was not the ones that I've mentioned, but they were very supportive of something called the Reber Plan. Now, Reber, who this was named after, was not an engineer. He was not a scientist. He was an impresario, sort of like Bill Graham, if the older ones remember Bill Graham Presents. He came up with the idea that you build a huge dam at the mouth of the Sacramento River at Suisun Bay, and this is going to create freshwater lakes. In other words, the idea, no water goes to the ocean. Any fresh water that goes to the ocean is wasted. And you still hear this idea floated around now. But the barrier of the dam, it had a rail on it. It had highway traffic. It reclaimed huge amounts of land. It had locks like the Panama Canal. It was absolutely a wild project. It was endorsed by the Hearst newspapers, like the Chronicle. The Corps of Engineers did a big study, and then the cost and the feasibility, they let it go. But you still hear this idea coming up over and over again. But the big thing that this Reber Plan did was it took 3 Berkeley faculty wives to found Save the Bay. Now, Kay Kerr, you probably have heard of the Clark Kerr campus. That was after Clark Kerr, the chancellor, and then the president. This was his wife, Sylvia McLaughlin's husband was the dean of engineering, and then the president of Homestake Mining, and Esther Gulick was very involved with her husband was on the faculty as well. And this Save the Bay was one of the major what they did was they realized the whole bay, if you look at from 49 to today, the whole bay was going to be lost. There was going to be concrete around it. They saw plans, started this organization, and were really very, very instrumental. And some of you may remember when they had when they were going to enlarge the stadium, and protesters were camping in the trees. They sort of very unfortunately got heard to Sylvia McLaughlin to pose. Not at that point of her life, it really wasn't fair that they had did that to her. But again, her legacy is really great. Luna Leopold, chief hydrologist at USGS, comes to Berkeley, 1972, has a long career here. I have to tell you, I know many of my colleagues got along great with him, but he scared the hell out of me. You know, I felt like I was a first-year graduate student whenever I went to talk to him, and he'd be quizzing me on things like that. But again, as time went on, he became really a very, 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 very nice colleague and even a friend. He was, of course, part of the famous Leopold family. And one of the nieces, Luna's niece, actually was a grad student for a while in forestry. I believe she was Reg Barrett's student. But of course, Stark Leopold, who was also, after Paul Needham died, was the director of Sage End Creek. He was one of the great wildlife biologists on the campus, and also a marvelous person. Alex Horn was one of the most interesting people on campus. He retired about 20 years ago. He came to Berkeley in the late 1960s to head up the Clear Lake Algal Research Unit. Clear Lake is the largest lake within California, and they had huge problems with algae that caused fish kills, and also a huge problem with this phantom midge, the Clear Lake gnat, that actually was they used DDE, a DDT derivative, to try to control it. And this is what led to the thin eggshells of the peregrine falcon, which were, really very, very important in Rachel Carson's expose. Alex, still active. He won the Best Paper Award for a topic he's been very interesting, to use aerators to solve water quality problems, especially algal blooms. And just a real character. He was on this task force about whether the Ebro River in Spain should have been diverted. And there's a kind of international group on a big pre-Zoom call, and Alex is getting ready to speak, and he brings out his guitar and he starts playing a song that he wrote about the Ebro River. He was just great, really a very, very interesting guy, and still around, and emeritus in engineering. Mary Power, did great things and is doing great things at the Eel River, the Angelo Preserve. Just many of you may not know, but Mary's first appointment at Berkeley was an ESPM as a visiting professor funded by NSF. And the movie based on the book, The Serengeti Rules, here you see Mary with the woman who played her as a young girl. One of the things that we lost, which was an incredible archive, was the Water Resources Archive. It was at Davis Hall, and it consisted of thousands of files that were about all student papers on Strawberry Creek, technical reports, things that the gray literature that was never online. But UC A&R, Ag and Natural Resources, took it over, didn't have the money to maintain it, and eventually moved it to Riverside, and a lot of the documents were lost. Field stations, you all know about the aquatic research that's gone on in the natural reserve system like nothing else in the world, but you may not know about the ag research stations like Hopland Field Station, Blodgett Forest. These are really excellent places to do research. They almost all have good aquatic habitats, UC Forestry Camp as well, the state parks, regional parks, and of course, even the Gump Research Station in Moorea, widely used for freshwater research, both by the class and by many of the researchers. I do want to mention one of the downsides about field stations in Berkeley, and that's the Bodega Bay Lab. Until 1988, Berkeley actually ran the Bodega Bay Lab. Now it's gone to Davis, it's been super successful. But one of the problems that Berkeley's had with field stations is they don't really take care of them. They don't support them. Very often they're not open to a wide variety of researchers. They become almost fiefdoms. And this is really what happened at Bodega Bay. I think the good thing to come out of the loss of Bodega Bay is we're very, very concerned now about keeping field stations, such as every time they talk about closing the gum station, somebody brings up Bodega Bay. And that's really something that's been very important. I want to end with, I'm talking about aquatic ecology at Berkeley, but one thing that really influenced us was the growth of aquatic ecology at Davis. 1958, Charles Goldman, who without a doubt the most famous limnologist in the world, goes to Davis. A year later, Davis becomes the second Berkeley campus. He sets up a lab at Lake Tahoe, and really limnology and aquatic ecology in California becomes identified with Goldman and all of his students. His best students as researchers in lakes all came out of his lab. And then in 74, Peter Moyle comes to Berkeley. Probably the most famous fish biologist in the United States right now. Jeff Mount, sedimentary geologist, gets converted into working on streams and water issues. Really amazing what Davis did. Of course, they have a fish biology department, just like Humboldt has a cooperative fisheries unit, but they have a center for watershed science. And, Davis has always been good. Maybe it's because they're right next to Sacramento and working on state problems. I mean, most of us would be more interested in working in Mongolia than like on Strawberry Creek. I remember one of the vice chancellors telling me when I first came here, you see that body of water out there, San Francisco Bay? It's like it doesn't exist to the Berkeley faculty. Nobody works on it. But Davis really does fill that niche. And I just want to conclude with one of the things that I'm very, very hopeful about is actually you look at the new aquatic ecologists. They're not going off to Mongolia. I mean, they're concerned with problems. And the fact that we have more aquatic ecologists at Berkeley, more graduate students than we've ever had before. And you look at Stephanie and her work, and Laura Larson, and Albert Rue, Ten Grantham, Robert Rue, Chris Martin, Evan Veriano, Paolo, our chair, Matt Condoff, who soon will be joining the retired ranks, and Tina Mendez. I think the future is great here. We're never going to, we have an opportunity. We have emphasis on California issues, which is going to help in the long run with funding. And like I said, I'm leaving my finishing seminar, as we use that term, really optimistic about the future. So good luck to all of you, and I hope we even get more, and thank you very much.
Well, thank you so much. There's a lot of virtual clapping going on, Vince. I realize some folks may need to jump off, and that's of course fine. And if Vince, do you have a little bit of time for answering questions? Is that okay?
A lot of time, yes.
Okay. So are there questions? And I apologize if I don't see. Oh, I do see a raised hand, but I have to find it. Well, yeah. Would people like to jump in? If you have a question, go ahead and jump. Unmute.
That's my raised hand.
Okay.
This is Gary.
Hi, Gary. Go ahead.
Good. Hey, Vince, how are you doing? Lovely talk.
Great to hear from you, Gary. Gary actually wrote with me and said, in an hour talk, at least half of it is going to be devoted to my research. So what are you going to talk about the other half?
Half was mine and half was Steve Balling's, I think. But anyway, the mention was enough. Lovely talk, and it's great to hear so much history of the Berkeley campus and the personalities and all that. Just kind of a quick question about Strawberry Creek. I couldn't help but wonder what we knew about Strawberry Creek prior to the campus, if anything, from old surveyors' information or anecdotal information. You mentioned the urban myth of steelhead in the river, and I'm kind of wondering whether in the 1800s, prior to campus, it did have a salmonid run that went up. Do we know much about that?
Yeah, it clearly did. Probably steelhead was the most common run, but I probably should mention this, but there was a settlement in the faculty glade Strawberry Creek, and they have found fish bones that were part of the diet, and presumably they're getting them from there. They're not bringing them up from the shell mounds that far, because this was a seasonal camp that they used. This is Kent Lightfoot has talked about that with us. So yeah, so I think that historically there were runs. Between the pollution, the culverting, I think that's what put an end to it. But thanks, Gary. Thank you. And Gary actually is one of the NSF directors right now. So.
And Justin and I cannot give you a grant, by the way, just in case you're wondering. So thanks a lot.
Thank you, Gary.
Yeah, are there other questions from the group? If not, I have Vince. I was wondering if we are reading so much and have been for the last 10 years about dam removal and about major restoration efforts. And I know, and I think Albert's work relates to this and other work about aquatic invertebrates as indicators of health and recovery. But what's you have a long perspective on both those dams and the potential outcomes of their removal, and I wonder if you could share your thoughts. Is that from your perspective, is it necessarily always a good thing?
Well, it's I see Allison Purcell is on. I was contacted by a foundation to come up with what suggestions would have big meaning, and I talked to Allison, and she talked about what we really need in terms of dam removal is good pre and post studies. We were involved with George Miller's attempt to take down Hetch Hetchy dam, and you could see that if you raised Don Pedro 25 feet, you'd have the same amount of water. But what's never really talked about is the recovery time. Probably, if you want Yosemite Valley again, you're probably talking about 100 years and a lot of engineering to get that done. I'm Allison is involved, and she's on. I don't know if she'd be able to speak, but with the Klamath River removal, which of course is going to be the really big one. And we've had small dam removals that look like they're going to turn out to be very, very successful. So, the big dam era in the United States is over. Where we're seeing it is in China and Africa, and maybe to a lesser extent in South America. But the work I did in Southeast Asia was looking at what's the effect of these large Chinese dams. Is it going to lead to schistosomiasis throughout the region, like it did when the Nile, when the Aswan Dam was built? What about migratory fish? What about the use of the floodplain, which your work showed how important that was, in the monsoonal rivers. So I think it's a good idea, but I think having some type of institute where this is studied before, during, and after would really be fine, because as you know, with monitoring, post monitoring is really hard to get funding for, and this I hope would be something that could be dealt with.
Absolutely. There are other questions? Vince here. Or just
Listen, this is Mike Peterson. I can jump in.
Yeah, go.
Hi Vince, this is Mike. Good to see you. I just was curious if given the history of mining in the state, like gold mining, if any of the earlier recent work looked at the legacy of mining on aquatic systems.
They've really done this in the Bay. This USGS has done this. The mining impacts that we would see, in Strawberry Creek down to the small estuary there, I think is going to be from Memorial Stadium, and I think separating those two is going to be very, very difficult. If I just point out, Mike is a wonderful example of what he's doing as he's one of the associate directors of the insurance agency in California, and I'm not pronouncing the agency correctly. But the reason is trained as an ecologist, what's the big issue in insurance right now? Floods and wildfires. So, having an ecologist as an associate director or assistant director is really very, very important. And it just shows what ESPM can do in terms of our graduates. We often think of, there's the academic route and that's it. But, Mike and many others, Bruce, who started his own consulting firm, Bruce Orr, really has shown what there's so many things out there that you can do a hell of a lot of good. And thank you, Mike.
Vince, this is Wayne Souza. I just wanted to thank you for a great talk. I learned so much. I've been on this campus for 44 years, and I learned so many new things in your talk about the history of the place. It's almost embarrassing. I wish I'd hung out with you more. But I do want to sort of second Justin's comments about you as a colleague. You were the first person to welcome me to the campus in 1977 outside of the zoology department. We were walking across campus and you just walked up and introduced yourself and gave me a hug, and that, the rest is history. And I really appreciate, the way you've been a fantastic colleague over the years, and I've loved interacting with you. So thanks very much.
Thank you. With Justin's comment and now Wayne's, I feel like I'm listening to an obituary.
That's right. I just retired too, so I'll join you on the other side.
Thank you. Thank you, Wayne.
Yeah, there are other comments. Thank you, Wayne, for that. And thank you, Mike.
So Vince, very nice.
Oh,
Been here the whole time. I wanted to follow on that. The mining question is of course, you spent some time in Mongolia. And I wanted to know if the type of mining contamination that you saw in Mongolia looks historically like California mining contamination, or is there any distinction? But also, of course, you've been a fabulous mentor and friend.
Matt is actually a physician working on snake bites and doing great work with that. Again, a former student in the lab. In Mongolia, it's quite different because what you have is artisanal mining, artisanal gold mining. And they're doing a lot of it at night. They're going mainly to abandoned mines and then using cyanide extraction. And actually, when I was there, I was trying to set up a monitoring program. I ended up getting cyanide poisoning, from doing that's not mercury poisoning from the cyanide. Because what happens is when you use cyanide, you're taking that off, and then the mercury that's left goes to methyl mercury, which is, as you know, is extremely toxic. Thank God I have a Chinese physician who pointed out that mercury is chelated by the Spanish parsley. And, so for 2 years, I was eating Spanish parsley 3 meals a day, but it did work. So thank you. Cilantro.
Yeah. Well, any last questions, comments?
Well, I'd like to thank everybody. There's so many of the former students that are on that, we're just spread out so much, and I really appreciate your coming on. And we will have Amy's making a recording for those that missed it. I know Stephanie and Mary weren't able to be here, but just to repeat, I'm really optimistic about the future for aquatic ecology at Berkeley, and maybe in 150 years we could hear another update. Thanks very much.
It's a fantastic way to end. Thank you very much, Vince, and thank you all for joining us today. And Vince, we're going to have lunch when you get back.
Okay, my treat.